Today we will talk about the central dogma of molecular biology. Central dogma of molecular biology. This central dogma of molecular biology, this concept is related to the flow of genetic information. So how the genetic information flows in a living organism, it is explained by means of central dogma of molecular biology. It explains flow of genetic information in living organisms. And this central dogma of molecular biology, it was proposed by Francis Crick. It was proposed by Francis Crick. The central dogma of molecular biology was proposed by Francis Crick, and this is related to the flow of genetic information. That means how genetic information flows in a living organism. So we have got DNA. DNA, it forms RNA, and RNA forms proteins. So we have got DNA forms RNA, it is called as transcription. Then RNA forms protein, it is called as translation. So RNA can be mRNA, rRNA, and tRNA. So the genetic information flows from DNA to RNA, RNA to proteins. This flow of genetic information in a living organism, it is known as central dogma of molecular biology. There was a scientist whose name was Teminism. He discovered reverse transcriptase enzyme in retroviruses. So this Teminism, he discovered reverse transcriptase. He discovered reverse transcriptase in retroviruses. What are retroviruses? Retroviruses are RNA viruses. Retroviruses, where RNA is the genetic material. So retroviruses are RNA viruses, where RNA is the genetic material. Here RNA is the genetic material. For example, we have HIV. In HIV, RNA is the genetic material. We have tobacco mosaic virus, RNA is the genetic material. We have coronavirus, RNA is the genetic material. So where RNA acts as a genetic material, those are called as retroviruses. This Teminism, he discovered reverse transcriptase enzyme in retroviruses. So in them, what happens? RNA synthesizes this. RNA synthesizes DNA. DNA synthesizes RNA, and RNA then synthesizes proteins. This is called as reverse central dogma. So in case of retroviruses, where RNA is a genetic material, this is for the retroviruses. Retroviruses, where RNA is a genetic material, the genetic information flows like this. We have RNA in retrovirus. RNA is the genetic material. So RNA will synthesize what? DNA. DNA will synthesize RNA, and RNA will synthesize proteins. This is called as reverse. This is called as reverse central dogma. Called as reverse central dogma. Reverse central dogma. A reverse central dogma. This was proposed by Teminism and Baltimore. Two scientists, Teminism and Baltimore. They proposed the reverse central dogma. Reverse central dogma means that RNA synthesizes DNA. This is called as reverse transcription. It's called as reverse transcription. Then DNA will synthesize RNA. This is simply transcription. And this is simply translation. This is simply translation. When RNA will synthesize proteins, translation. And the main enzyme in reverse transcription is reverse transcriptase. The main enzyme in reverse transcription, that is reverse transcriptase. This reverse transcriptase, it is basically this reverse transcriptase. This is basically an RNA dependent This is also known as RNA dependent DNA polymerase. DNA polymerase. This reverse transcriptase enzyme is also known as RNA dependent DNA polymerase, and this reverse transcription is also called as Teminism. This reverse transcription is also called as Teminism. So what do you mean by Teminism? Teminism is simply the reverse transcription. Reverse transcription is also known as Teminism. This occurs in case of retroviruses, where RNA acts as a genetic material. This is what is called as reverse central dogma. Now, because of the fact that in case of retroviruses, RNA is a genetic material, now this central dogma, this was the first central dogma which was proposed by Francis Crick, that DNA gives RNA, RNA gives proteins. This was the earlier version of the central dogma of molecular biology. Then was discovered retrovirus, reverse transcriptase, where RNA was found as a genetic material. Now the modern version of the central dogma of molecular biology is, this is modern version of central dogma of molecular biology. Modern version of central dogma of molecular biology. It is like this. We have DNA, we have RNA, and we have what? We have proteins. So DNA, it will give rise to DNA. This is called as replication, DNA replication. This is DNA replication. When DNA will synthesize its own copies, this is called as DNA replication. Then DNA can synthesize RNA. It is called as transcription. And main enzyme in transcription is RNA polymerase. Then this RNA can synthesize DNA. This RNA can synthesize DNA. This is what is called as reverse transcription. Reverse transcription. The main enzyme in reverse transcription is reverse transcriptase, and this reverse transcriptase, it is an RNA dependent DNA polymerase. As we have to synthesize DNA, so it is DNA polymerase, but it depends upon the RNA. So it is called as RNA dependent DNA polymerase. So this is called as reverse transcription, and this reverse transcription is also known as Teminism, as it was first described by Temin. Then we have RNA. RNA can synthesize its own copies, what is called as RNA replication. This is RNA replication. Then RNA can get involved in the formation of proteins, that is known as translation. This is what is the modern version of central dogma of molecular biology. That means how the genetic information flows in living organisms. It was first proposed by Francis Crick. It was simple: DNA gives RNA, RNA forms proteins. But with the discovery of the retroviruses, reverse transcriptase, it was a little bit modified, and nowadays we have got a modern version of this central dogma of molecular biology, where we have got DNA. It will form its own copies, that is called as DNA replication. DNA will form RNA, that is called as transcription. Main enzyme is RNA polymerase. This RNA can synthesize DNA. It is called as reverse transcription or Teminism. The main enzyme is reverse transcriptase. It is called as RNA dependent DNA polymerase. Then RNA can form the protein, that is called as translation. This is the modern version of the central dogma. So this was we talked about the retroviruses, or those viruses where RNA as a genetic material. Then in them reverse central dogma takes place. Thanks.